Alrighty, everybody, in this third part of videos for the autonomic nervous system, we're going to talk about some neurotransmitters. And the word neurotransmitter shouldn't be totally unfamiliar to you guys. We've talked about neurotransmitters before. Specifically, we talked a lot about acetylcholine at the neuromuscular junction. But in the autonomic nervous system, we have a couple of major neurotransmitters, and those major neurotransmitters are acetylcholine and norepinephrine. Okay, so major neurotransmitters, acetylcholine and norepinephrine. In terms of the nerves and which nerves release which neurotransmitters, we give them names. And so neurons that release acetylcholine we call cholinergic fibers. And in the autonomic nervous system, all preganglionic axons are cholinergic, and all parasympathetic postganglionic axons are cholinergic. So as you can see over here, all preganglionic axons are cholinergic because they're all three preganglionic neurons are releasing acetylcholine. And then all parasympathetic postganglionic axons release acetylcholine. So all postganglionic axons or neurons in the parasympathetic nervous system are cholinergic. Okay? The other type of fibers that we'll talk about are called adrenergic. And if you need help remembering this, you think of adrenergic as like adrenaline, because they release norepinephrine, and epinephrine and norepinephrine are the fancy words for adrenaline. So most sympathetic postganglionic axons are adrenergic. Okay, so here we see this postganglionic axon releasing norepinephrine. And there would also be, this is kind of a special case in the adrenal gland, that the adrenal medulla directly releases epinephrine and norepinephrine. And so it's got some adrenergic effects. The exceptions for this postganglionic axons being adrenergic for the sympathetic nervous system, the exceptions are sympathetic postganglionic fibers that are going to sweat glands and also some blood vessels in skeletal muscles. They will actually be cholinergic. Okay, so exceptions. These sympathetic postganglionic fibers that are going to the sweat glands and to certain blood vessels and skeletal muscles are actually cholinergic. Okay. Now, the effect that this neurotransmitter is going to have, it is going to depend on whether it binds to a cholinergic or an adrenergic receptor, and also what type of cholinergic or adrenergic receptor it is, because we have several types of cholinergic and adrenergic receptors. So first, let's talk about cholinergic receptors. And let's also remember that cholinergic receptors are the ones that are releasing acetylcholine. We have two types of cholinergic receptors. We have nicotinic and muscarinic receptors. And these two types, they're actually named after drugs that bind to these receptors and mimic the effects of acetylcholine. And those drugs would be nicotine, would bind to nicotinic receptors, and they mimic, they do the same thing as what would happen if acetylcholine binded to that receptor. And then muscarinic receptors actually are named after the mushroom poison muscarine. And when muscarine binds to these receptors, these muscarinic receptors, the same thing happens as what would have happened if acetylcholine had bound to that receptor. Okay, so that's just how they're named. In terms of these receptor types, I believe I am going to go into more detail on the next couple of slides. Yes, I am. Okay, so this is just a brief overview. In addition, I also have this table over here, and then sort of just this summary table. So these are good tables to refer back to just to get sort of a general understanding of what everything is doing and what everything does. And then we'll talk a little bit more about these drug classes that influence each of these different receptors. All right, so first let's talk about nicotinic receptors. Nicotinic receptors are going to be found on all postganglionic neurons of both sympathetic and parasympathetic neurons. So all the postganglionic neurons in the autonomic nervous system are going to have nicotinic receptors. In addition, the hormone-producing cells of the adrenal medulla will also have nicotinic receptors. And the sarcolemma in the skeletal muscle at the neuromuscular junction, this should not be anything new, will also have these nicotinic receptors. Okay, so as I mentioned, I just paused the video so I could draw this quick little tidbit for you just to kind of illustrate what I mean by these receptors. So these nicotinic receptors, they're on all postganglionic neurons, both sympathetic and parasympathetic. And so that's what we see kind of going on here. So here's our preganglionic neuron, and it's going to synapse onto the cell body of this postganglionic neuron, right? And say this is the parasympathetic nervous system. The cell body of this postganglionic neuron is going to have a nicotinic receptor so that when the presynaptic neuron comes and releases acetylcholine, that acetylcholine is going to bind to that nicotinic receptor and is going to excite the postsynaptic, or excuse me, the postganglionic neuron. Okay. A very similar situation would be occurring down in the sympathetic nervous system, where the preganglionic neuron is going to release some acetylcholine, and the cell body of the postganglionic neuron in the sympathetic nervous system is going to have a nicotinic receptor on that cell body that's going to bind that acetylcholine and thus cause excitation of this postganglionic neuron in the sympathetic nervous system. In addition, this is meant to represent the adrenal medulla. The sympathetic nervous system preganglionic neuron is going to come in and synapse onto hormone secreting cells that are located on the adrenal medulla, and those hormone secreting cells actually have nicotinic receptors. All right, so these two little blue lines are meant to represent those nicotinic receptors, and that's going to excite these hormone producing cells in the adrenal medulla. And actually what's going to happen in terms of the adrenal medulla is it is going to, these hormone secreting cells are actually going to be excited, and they are going to release epinephrine. Okay. In addition, we'll also find nicotinic receptors on the sarcolemma of skeletal muscle cells at the neuromuscular junction. So that's kind of what I tried to draw here and represent, this yellow being the neuron. Here's our muscle cell, and we know from our studies that when the neuron is excited, it's going to kick out this acetylcholine that used to be packaged inside of vesicles, right? So that fuses, and then we kick out the acetylcholine. And then the muscle cell has receptors on it that the acetylcholine will bind to, and that's going to cause excitation of the muscle cell through the opening and closing of different sodium and potassium channels, which causes depolarization and muscle shortening ultimately. Okay, so the effect of acetylcholine at nicotinic receptors is always stimulatory. So it always results in opening ion channels and causing depolarization at the postsynaptic cell. Okay, whether that be a postganglionic neuron, a hormone producing cell, or a muscle cell. It's always stimulatory. So that's what you see here in this table. Nicotinic receptors are located on all postganglionic neurons, also on adrenal medullary cells, and also at the neuromuscular junctions of skeletal muscle. And the effect of acetylcholine binding to nicotinic receptors is always excitation. Okay. So now let's talk about the other type of cholinergic receptors, which are called muscarinic receptors. And I'm going to just clear all my drawings here. The muscarinic receptors are found on all effector cells that are stimulated by postganglionic cholinergic fibers. And so this would be all of the parasympathetic target organs. Okay, so all of our parasympathetic target organs are going to have these muscarinic receptors. And addition to all of our parasympathetic target organs, there are some sympathetic target organs that will have muscarinic receptors. And muscarinic response, the effect of acetylcholine binding to the muscarinic receptors, can either be stimulatory or inhibitory. It depends on the receptor type of the target organ. So in terms of our parasympathetic target organs, most of the time whenever acetylcholine binds to a muscarinic receptor, it will result in excitation. However, if acetylcholine binds to a muscarinic receptor that's on the cardiac muscle, it's actually going to inhibit the cardiac muscle. This would cause things like a decreased heart rate. In addition, as I mentioned before, there are limited sympathetic targets, for example, certain sweat glands. And if acetylcholine is released onto, for example, muscarinic receptors that are located on sweat glands, this activates the sweat glands, because remember, sweat glands are only innervated by the sympathetic nervous system. And this acetylcholine will be binding onto muscarinic receptors on those sweat glands. And so this probably sounds kind of confusing when I'm just sitting here explaining it. So let's just say, let me just do it like this. Okay, pretend this is our sweat gland. All right, and we have little sweat molecules inside of here. And we're going to say that we have, here's our sympathetic preganglionic neuron, and then we're going to have our sympathetic postganglionic neuron. And for this particular example, we're going to say that our sweat glands have muscarinic acetylcholine receptors. And so whenever this muscarinic receptor is bound to acetylcholine, or acetylcholine comes and binds onto that muscarinic receptor, this is going to cause the sweat gland to activate. Okay, and so then that sweat can be released. Okay, hopefully that makes more sense. All right, so this is a good little video that I'm going to play for you guys about the different receptor types. We're going to start it at 2:06.
There are two broad classes of ACH receptors, nicotinic and muscarinic receptors. These receptors are so named because of the drugs nicotine and muscarine that selectively activate each of the receptors. Most nicotinic receptors are ionotropic, responding rapidly and usually having an excitatory effect. The drug curare, once used as an arrow poison by South American Indians, blocks nicotinic ACH receptors. Because the synapses between nerves and skeletal muscles are nicotinic, curare paralyzes all skeletal muscles, including those used in breathing. Muscarinic receptors are G-protein coupled metabotropic receptors, so they have slower responses when activated, and they can be either excitatory or inhibitory. Muscarinic receptors can be blocked by the drugs atropine or scopolamine. Certain organophosphate insecticides, such as diphenyltrichloroethane, DDT, and the nerve gas sarin, inhibit the enzyme acetylcholinesterase, causing ACH to accumulate at the synapse. This buildup of acetylcholine depolarizes the postsynaptic cell and renders it refractory to subsequent acetylcholine release, causing, among other effects, neuromuscular paralysis. Many other drugs also act at cholinergic synapses. For example, botulinum toxin is a potent bacterial poison that can grow in improperly canned food. Botulinum blocks the release of acetylcholine, resulting in muscular paralysis and asphyxiation. In much diluted form, botulinum toxin is marketed as Botox, which inhibits facial wrinkling by locally paralyzing facial muscles. Tetanus toxin is chemically similar to botulinum and is responsible for tetanus poisoning, lockjaw. Finally, black widow spider venom causes the continuous release of acetylcholine at the neuromuscular junction, causing the muscle to respond violently until the supply of acetylcholine is depleted.
All right, so that was just a quick little video. I just thought it was interesting, and so I wanted to share that with you guys. And now we're going to move on and talk about adrenergic receptors. So remember, adrenergic, I gave you guys a hint earlier, but I will say it again. Adrenergic, think of adrenal, which would be norepinephrine and epinephrine. So we have two types of adrenergic receptors. We have alpha type, and alpha has two subtypes. So there's a alpha 1 and an alpha 2 subtype. And then we have beta type, and beta has 3 subtypes. We have type 1, 2, and 3 beta. And the effect of binding these receptors, it just depends on which subclass of receptor predominates on the organ that is being targeted. So for example, if we are looking at, let's say, cardiac muscle, when norepinephrine binds to cardiac muscle on beta 1 receptors, it might cause an increase in heart rate. Okay, so this would be a stimulatory effect if we're talking about the heart. However, epinephrine or norepinephrine, this adrenaline, whenever it binds to this beta 2 receptors on the lungs, this causes relaxation of the lungs, and that would be an inhibitory effect. So again, it just depends. The effect just depends on what type of receptor we're talking about, and also it depends on what organ you're talking about. So let's look at each of these subtypes in a little bit more detail, because I think this is very useful stuff to know. So for example, beta 1 adrenergic receptors are commonly located, they're predominantly located in the heart, but they're also located in the kidneys and adipose tissue. And so whenever norepinephrine or epinephrine binds to beta 1 receptors, it's going to cause an increase in heart rate and also cause the kidneys to release renin, which will go on to increase blood pressure. Okay, so big thing here is increasing heart rate and stimulating the kidneys to release renin, which I'm just going to write this in here because it's something that you should know. Renin increases blood pressure. Okay. If we look at beta 2 receptors, beta 2 receptors are located in the lungs and most of the other sympathetic organs. They are very abundant on blood vessels that serve the heart, the liver, and the skeletal muscle. And whenever beta 2 receptors are bound by norepinephrine or epinephrine, mostly the effect is inhibitory. And so that would be, if it's inhibitory, that would be things like relaxing the blood vessels that are on those locations, right? So this is majorly inhibitory. Beta 1 is majorly stimulatory. We have beta 3 receptors located on adipose tissue, and the effect of these beta 3 receptors being bound by norepinephrine. So if epinephrine and norepinephrine come in and they bind to these receptors, this is going to have a stimulatory effect. It's actually going to stimulate lipolysis. So it's going to facilitate the breakdown of fat cells. Virtually all sympathetic target organs, especially blood vessels that serve the skin, the abdomen, the kidneys, and the salivary glands, basically all those blood vessels have alpha-1 receptors. And there's also alpha-1 receptors on the pupils. And so this is an instance in which the same receptor would have opposite effects. So in terms of the blood vessels and the visceral organ sphincters, it would constrict them, so that's a stimulatory effect, but it's actually going to dilate the pupils, right? So that's actually an inhibitory effect. Okay, so again, this just goes back to the idea that even though it's the same receptor, it can have a different effect depending on which organ you're talking about. And then finally, these alpha-2 receptors are going to be present on the membranes of adrenergic axon terminals, and they're also going to be present in pancreas. And so what we can see from the table here is that alpha-2 receptors are primarily inhibitory receptors. They inhibit the release of norepinephrine and also inhibit the release of insulin from the pancreas. Okay. There are certain drugs that we pharmacologically find very useful because we know so much about these receptors. And so we have these sympathomimetic agents and sympatholytic agents. So sympathomimetic is going to mimic the sympathetic nervous system. Okay. These are things like albuterol. If you have asthma, maybe you have heard of albuterol. Albuterol is a medicine that they put in inhalers, and it actually will help, and if you're having an asthma attack, it will help open up your bronchioles because it's actually going to bind to beta-2 adrenergic receptors. So we have beta-2 adrenergic receptors located on the lungs, and it's going to remember beta-2 receptors in the lungs dilate the bronchioles. And so albuterol is something that we can put in medication form that when you take it, it will then dilate the bronchioles. And the same sort of situation here with phenylephrine. Basically, this is the human-made form of epinephrine, of adrenaline. And this is something that you'll find in a lot of cold medication. It is a nasal decongestant, and it actually binds to these alpha-1 receptors. And this is actually very useful because if you are stuffed up, if you're stuffy, a lot of times it's because the vessels in your nasal mucosa are dilated. And so by binding to these alpha-1 receptors, it actually constricts your blood vessels. And by constricting the blood vessels, that actually will cause you to have decreased congestion because there's less blood in your vessels in your nasal mucosa. All right, so that's how a lot of cold medicines work, as nasal decongestants. The other drug class here are these sympatholytic. So basically, these particular drugs are going to be ones that do the opposite effect of the sympathetic nervous system. Okay, and so the example given here is called propranolol, and propranolol happens to be what we call a beta blocker. And beta blockers are given to people that have high blood pressure. And so basically the way propranolol works, and actually anything that ends in the suffix OL, is going to be a beta blocker. And so propranolol is a beta blocker because it blocks these beta receptors. And so by blocking these beta receptors, you are going to, specifically, if you take a look at, I'm so sorry, apologies. Specifically, if you take a look at these beta-1 receptors, remember the beta-1 receptors, if they are bound what's going to happen? Your heart rate is going to increase and your blood pressure is going to go up. So if you block these beta-1 receptors, then your heart rate will go down and your blood pressure will go down. So these have become very, very useful drugs to treat things like hypertension. So I've already covered then these two things. I'm now just going to briefly talk about the drugs that went with acetylcholine. So these are either nicotinic or muscarinic receptors. Let's talk about, these are epi, norepi. So these are alpha or beta receptors. So let's talk about up here these drugs that can influence either nicotinic or muscarinic receptors. So first we'll talk about nicotinic agents, such as nicotine, for example. This is actually one of the ways that we clinically use this is by putting it into products that are used for like smoking cessation. So nicotine patches, nicotine gum, that kind of stuff. And what actually, how this works is that it binds to these nicotinic acetylcholine receptors and will stimulate sympathetic effects like blood pressure rising. And you learn this, and this becomes a learned response to help you quit smoking. These parasympathetic mimetic agents, these are also called muscarinic agents. They bind to muscarinic acetylcholine receptors and enhance the parasympathetic activity by mimicking the effects of acetylcholine. So things like pilocarpine, bethanchol. So pilocarpine is used for treating glaucoma because it is able to open the drainage pores, which is a relaxation effect of parasympathetic nervous system. Additionally, in the parasympathetic nervous system, we know this is our rest and digest, and so you're able to, in this system, in the parasympathetic nervous system, you would be doing things like urinating. But if you have difficulty urinating, you can take this drug, this one, and this actually will increase bladder contraction to help you get past the difficulty urinating. Additionally, the final class here are called acetylcholinesterase inhibitors, and these ones actually don't bind to the receptor, but they do bind to what's called acetylcholinesterase, which is the enzyme that basically takes away acetylcholine from the synaptic cleft, and so you can end the effect of acetylcholine on the receptor. And so one of the ways that we can use acetylcholinesterase inhibitors, just gonna draw something out for you guys really quick. Okay, so I just drew on here a little example of how this and why this works, because sometimes this can be a little bit confusing. So remember, at the neuromuscular junction, that's what you see here drawn, we have this acetylcholine packaged in vesicles, and then whenever the nerve gets stimulated, we spit out this acetylcholine. Acetylcholine goes over to the muscle cell, and it excites it. right? Now, normally in order to end this muscle contraction, what's going to happen is that we're going to have acetylcholinesterase come in and pluck off these acetylcholine molecules. So we're plucking those off. And then it's going to chew up the acetylcholine and give it back to the neuron. Okay, so that would make all this space clear. And that would thus end the muscle contraction. Or if this was a postsynaptic neuron, it would end the stimulation of that secondary neuron. Now, how acetylcholinesterase inhibitors work is they basically just block the activity of the acetylcholinesterase. And so therefore, all of the acetylcholine is going to stay bound and continue stimulating, right? We're going to continue stimulating here for as long as possible. And this is really useful in diseases like myasthenia gravis, because in myasthenia gravis, the problem tends to be there's not enough acetylcholine, and this leads to like muscle instability and so on. And so by keeping acetylcholine bound to its receptor for just a little bit longer, you're able to provide more smooth muscle movement. The other example here is what's called sarin. And sarin is sort of similar to insecticides, but sarin gas was used as a chemical warfare agent, would cause the same effects as this acetylcholine esterase inhibitor does for myasthenia gravis. Okay. So now let's talk about these localized versus diffuse effects of autonomic fibers. So in terms of the parasympathetic division, parasympathetic effects are very short-lived and they're highly localized over the effector tissue. Okay, so the responses don't last very long and they're very localized to just that space. However, the sympathetic response, the sympathetic effect, these are very long-lasting effects that are body-wide. And this is because norepinephrine and epinephrine actually get released into the blood, and they will stay there until they're destroyed by the liver. Norepinephrine, so I keep saying norepinephrine and epinephrine, and just to kind of clarify that, norepinephrine is going to have the same effect as what sympathetic neurons have in the blood, but epinephrine is more potent at increasing heart rate, raising blood glucose, and also raising the metabolic rate. So which one's stronger? Epi is stronger than norepinephrine. Okay.